Morning ma'am. Good morning everyone. I am from second year CZPT combination. Today one of my friend Meghana from CVPT combination is presenting presentation on the topic of carbohydrates. For the presentation.
Good morning one and all. So today my topic is about carbohydrates. So let us know about the sources of carbohydrates, types of carbohydrates and other things. Let us know about the introduction of carbohydrates. First, definition. Carbohydrates are organic compounds made up of carbon, hydrogen and oxygen, usually in the ratio of 1:2:1. That is carbon, hydrogen and oxygen. Next, general formula. Most carbohydrates have the formula CH2O. That is, C is having the ratio of 1, hydrogen is having the 2 ratio and oxygen in the 1 ratio. Next, main function of carbohydrates. They are the primary source of energy for living organisms. Next, they are the building blocks and simplest carbon of monosaccharides like glucose and fructose. In carbohydrates, we can see the simple carbohydrates and complex carbohydrates. Simple carbohydrates are the example of. In simple carbohydrates, we can see glucose, fructose and galactose. And coming to the complex carbohydrates. Complex carbohydrates are the combination of monosaccharides. These are the monosaccharides. Combination of monosaccharides are called as complex carbohydrates. For example, starch. You already know that starch is a storage compound of plants. Next, examples. Okay, I have already told glucose, sugar in the blood, sucrose, table sugar, starch in plants, cellulose is present in the plant cell. Next slide. Okay, coming to the sources of carbohydrates. Common carbohydrate sources are grains, bread, pasta, cereals and crackers, fruits. See, carbohydrates can be, as it is a primary source, it could be seen in very much amount, and in the lipids there will be a less amount of carbohydrates as proteins. See, glycoproteins, lipoproteins and glycolipids. With the lipids, carbohydrates are present. With the proteins, carbohydrates are present but in a small amount. Next slide please. Classification of carbohydrates. Simple carbohydrates, complex carbohydrates. In the simple carbohydrates, we can see two types, that is monosaccharides and disaccharides along with oligosaccharides. Next, complex carbohydrates, you can see polysaccharides. Poly, the name itself says many. Many means large amount of monosaccharides combined together to form a polysaccharides. Next, in the monosaccharides, we can see glucose, fructose, galactose. And disaccharides. Disaccharides mean combination of two monosaccharides is called as disaccharides. For example, maltose, lactose and sucrose. Next, can anyone say maltose is a combination of lactose is a combination of galactose. Next, carbohydrates. As I said, carbohydrates are made up of carbon, hydrogen and oxygen, which C6H12O6. Next, forming ring structure. Can anyone know the ring structure of glucose? This is the ring structure of glucose. It is also called as Harvard structure. Next, polysaccharides are formed by linking many monosaccharides through glycoside bonds. This is very important. Monosaccharides combine together to form polysaccharides. How they are bonded? Through what the linkage? Glycoside linkage. Either alpha linkage or beta linkage. Next, the structure determines the function. Starch for energy and cellulose for the structure. Cellulose is a very important compound which is a strong structure which cannot be digested by the human beings. And starch. Starch is stored for the energy in the plants. Next. See, this is the structures. You can see glucose, ribose. Coming monosaccharides, disaccharides, and coming to the polycomplex carbohydrates, that is polysaccharides. These are the glycosidic linkages. Next slide, please. Next, coming to the properties of carbohydrates, there are two types: physical and chemical properties. Coming to the physical properties, they are sweet in taste. Generally, they are very much soluble in water. Unlike lipids, lipids are not soluble in water because some organic without organic solvents they are insoluble. With organic solvents, some lipids are soluble. Next, colorless and crystalline. See, the carbohydrates are sweet in taste. Generally, it is not sweet in taste because of the saliva. They mix with saliva gives the sweet taste. Next, polysaccharides like starch, cellulose are tasteless and insoluble in water because of the linkages. They are very hard and couldn't be breakable easily, so they are insoluble in water. They can form viscous or gummy solutions. Starch paste, for that's example. And coming to the chemical properties, reducing property. First is reducing property. But as some sugars like glucose reduce Felix or Benedict's solution, it can be done in the laboratory. And such sugars are also called as reducing sugars. Second one, oxidation. Carbohydrates can be oxidized to acids. For example, glucose convert into, for example, I show you CH4, CH, CH2. See here, the aldehyde group, the addition of oxygen takes place. It forms the glucose converted into the glucose can be converted into acid, that is gluconic acid, with the addition of oxygen. Next. Third one, dehydration. When heated with acid, that is carbohydrates can be heated with acid, they use water to form caramel or purple compounds. Fourth one, fermentation. Sugars like glucose are converted into alcohol and CO by yeast. Fifth one, hydrolysis. Complex carbohydrates, that is starch, sucrose, can break down into simple sugars by Ribose and deoxyribose sugars are part of RNA and DNA. Ribose, where is ribose is present? RNA. Deoxyribose?
DNA.
Because when ribose loses one oxygen in a Haber structure, there will be a OH, that is ribose. When it loses one oxygen, it becomes deoxy. Loses of oxygen forms a deoxyribose that can be seen in DNA. Next. Yeah, sweetening agent. Sugars like glucose and sucrose gives sweet taste to food. Self-recognition and communication. Present on cell membranes helps in cell signaling and recognition. Coming to the concept of monosaccharides, let us know the introduction. It is, as you all know, simplest form of carbohydrates. General formula. Okay, can anyone fill up that?
CH2.
COOH. Okay. Cannot be hydrolyzed into simpler molecules. That is, polysaccharides can be hydrolyzed into monosaccharides, but monosaccharides cannot be hydrolyzed because it is the main simple component, smallest components that couldn't be hydrolyzed. Next. Example: glucose, fructose, galactose, building blocks of disaccharides and polysaccharides. Next. Isomerism and monosaccharides. What is meant by isomerism? Same molecular formula but different structural formula. So let us see isomerism in monosaccharides. Same molecular formula but different structure. Types of isomerism: structural isomerism, stereoisomerism, arrangement isomerism. Okay. Isomerism, different atoms arrangement, glucose and fructose. See, the general formula of glucose and fructose is same, but the structures of glucose and fructose is different. That is the structural isomerism. Coming to the stereoisomerism, different spatial arrangement can be seen. That is real end forms. That is dextro and levorotatory structures can be seen. And based on the position of OH, epimers, different at one carbon, that is glucose, for example, glucose and galactose. Anomers, different at anomeric carbon, that is alpha and beta forms. See, you can see the structure. Open structure, ring structure. This is the enantiomers, that is D-glucose and dextro and levorotatory glucose. And here you can see the glucose and galactose, how it is differentiated as epimers. Only one chiral carbon, different configuration. Next, anomers, how it is different. Upside down with the anomers, opposite are the epimers. Next, ring structure of monomers. Monosaccharides mainly exist in cyclic forms, as you all know, glucose and fructose, formed by reaction between aldehyde and ketone group and an OH group. Types of rings: furans, furanose, 5-membered ring, that is fructose; pyranose, 6-membered ring, example glucose. And there will be two forms of structures, that is alpha and beta anomers. Next. You can see the structure. Glucose, fructose. 6-membered rings are also called as hexose. 5-membered rings are also called as pentoses. Next, sugar derivatives of monosaccharides. There are 5 types of sugar derivatives. That is sugar alcohols, reduction of aldehyde. Next, sugar alcohol, that is glucose on the aldehyde reduction convert into sorbitol. That is, the ketone group is converted into a alcohol group. That is, reduction of aldehyde or ketone group into alcohol. This is the sugar alcohol. Next, amino sugars is also one of the type of sugar derivative of carbohydrate. For example, glucosamine. Glucose is converted into glucosamine. How? See here, the one OH is converted into, as the name itself suggests, amino. One OH group deoxy sugars, as I said, ribose is converted into deoxyribose by losing one oxygen. See here. OH group is reducted to hydrogen. Next. Sugar acid is also one of the type, that is glucose is converted into gluconic acid. As I told here. Carboxyl group again convert. That is the addition of oxygen. Glucose is converted into gluconic acid. And the next type is phosphate sugar, that is the addition of phosphate group to the glucose. So you can see PO4. This is the CH4 group, C will be PO4 add up. That is the phosphate sugars. Next. Oligosaccharides. Oligosaccharides are made up of many 2 to 10 monosaccharides. The types of oligosaccharides are disaccharides, tetrasaccharides, etc. And coming to the oligosaccharides are the polymers of monosaccharides containing 2 to 10 residues accumulate in vacuole. While polysaccharides in plastics are classified as disaccharides. Yield 2 monosaccharides from hydrolysis. Like yields 2 monosaccharides from hydrolysis. It has 2 types, reducing disaccharides and non-reducing disaccharides. Example, maltose is combination of glucose and glucose, lactose, glucose plus galactose, and other examples. For the non-reducing disaccharides, sucrose, glucose plus fructose. And the next, trisaccharides means 3 monosaccharides used on hydrolysis. Next, raffinose is an example for trisaccharides. It is the combination of glucose, fructose, and galactose found in cottonseed and sugar. Tetrasaccharides yield 4 monosaccharides on hydrolysis. Example, stachyose, glucose, fructose, galactose, and galactose. So it is an example of tetrasaccharide. Coming to the sucrose. Okay, sucrose is a kind of sugar. Sugar. What type of sugar? Is a type of what sugar? Yes. That is under disaccharides. Next slide. Okay, it is a disaccharide made up of glucose plus fructose. Chemical formula of sucrose is C12H22O11. Next, linked by alpha 1,2 glycoside linkage. This is the alpha. This is the alpha 1,2 alpha C alpha 1,2 linkage, that is glycosidic bond can be seen. And next it is a non-reducing sugar, as you all know, sweet, crystalline and water soluble. Polysaccharides are insoluble. This is the water soluble. And on hydrolysis leaves glucose and fructose, that is invert sugar, which means the sucrose is converted into glucose and fructose is the inversion of sugar. Next, 8 point, it is also used as the table sugar and an energy source. Next, fructose. Coming to the fructose, it is a monosaccharide ketohexoses. Chemical formula C6H12O6 found in fruits, honey and corn syrup. As a ketone group at carbon, forms a 5-membered Fernandez ring 6. It is a reducing sugar. Sweetest among the natural sugars, provides quick energy to the body. Coming to the polysaccharides, many monomers combine to form the polysaccharides are the complex carbohydrates made up of many monosaccharide units joined by glycosidic bonds. For general formula C6H10O5, they are the macromolecules usually insoluble in water and non-sweet compared to the glucose and fructose. Next, there are 2 types, homopolysaccharides and heteropolysaccharides. Homo, which means the same, hetero, ಮಾಡಿದಾಗ ಈ ಕಲರ್ಗಳು ಬರುತ್ತೆ. ನೆಕ್ಸ್ಟ್ ಸ್ಟೋರೇಜ್ ಸೈಟ್, ವೇರ್ ಡು ದ ಸ್ಟಾರ್ಚ್ ಫೌಂಡ್. Storage, seeds, tubers and glycogen found in, as it is animal storage, it is found in liver and muscles. Next, structure and reaction of starch. See, alpha carbon 1,4 glycosidic linkage and upside down that is 1,4 1,6 linkage. Next, hydrolysis. Starch on hydrolysis gives glucose and fructose. Next slide. Amylose. See, amylose is a single chain that is simple, and amylopectin is a branching structure. Properties of starch. Starch are white, tasteless, and odorless powder. Insoluble as it is it is insoluble in cold water and forms a paste. When heated with water, gives blue color with iodine solution. On hydrolysis, gives maltose and glucose. Burn to produce carbon dioxide and water and converts into dextrine on strong heating. Enzyme amylase breaks into maltose proteins, plain polarized red. Serves as main energy storage material in the plants. Starch structure.
Ayo.